That works. Okay, thank you so much for having me here and giving me the chance to present. So I wanted this talk to be a bit about like the career path. I guess that's part of it. So, but I also wanted to teach the tools that we have developed. So I'm kind of like try to stitch that together. Like what was that tool at that time in my career path, and like why was that important later on. Before getting started, I wanted to introduce myself. I initially wanted to really write down my whole educational path because it's a bit unique and very uncommon. I didn't go a very straight path. I went through the school in the German system that didn't allow me to go to university. I was told I was not allowed to work on computers, not allowed meaning I have no chance and so on. But at the end, I somehow made it to university. That happened like quite late on. And when I started when I was 25, I guess, with university. So I wanted to just say these words in the beginning. So it doesn't matter where you come. You can always go to university. You can always make it. You can always do great work. So I'm born in a small city in Germany. It's called Erding. Some of you might know that there's beer from this region. It's called Erdinger. You might have drinking that at one point. And I did go to the university in Munich, LMU and TU Munich. Normally at that point, I explained what Burkhard Rost is doing. I think many people of you might have heard about Burkhard Rost here. He was a big important part for the organization. But I guess I want to say what Burkhard Rost was for me. Burkhard Rost was for me getting a bit rid of my imposter syndrome. So he encouraged me to be a scientist. So as was during my undergrad studies, he saw potential and he said like, oh, that is great. You should go on. You should do your thing. I think you will do fine. So I did do my bachelor's with him, working on in silico prediction of mutations. Then I was always curious to see more places. So I went to André Charlet at UCSF for an internship that was in San Francisco. So that was really exciting that I had the chance to see one of these, like, I guess, prestige universities, because I was always curious, what is a prestige university? Are they like magically better? But I realized they are good, but they're not so different. You know, they are not so different than the universities I've been to and the institutions I have seen before. Andre doing great work on structures. So I was working on structures there and see this integrative modeling world. I then did my PhD with Johannes Söding, and he was in the Max Planck in Göttingen. And I didn't spend a single day in Göttingen, actually. So I did my PhD with Johannes. And I worked really intensively with him, but I did not spend the same I didn't share the same physical space. I was at Barcelona at the CRG. If you want to go to an institute that has a very nice beachfront, the CRG is the place to be. So I worked with Cedric, invited me to be in his lab, and so I was there, and I could work on my project, but I also could see what happens in Cedric's lab, and they were like building up these Nextflow pipeline, Nextflow systems. I could see what they do with multiple sequence alignments. So really exciting that I had the chance to be there. And then I decided I wanted to go out, and I wanted to go a bit further. So for the rest of my PhD, I was at Seoul National University with Sok Chaok. She works in the chemistry department on structure prediction, and she was so kind to give me a space. I was initially wanted to only stay for 1 year. I ended up staying 3 until the end of my PhD. Then I moved on and went to Stephen Salzburg at Johns Hopkins University. So Stephen really known for a lot of like genomics tools. And I wanted to see a bit more of the genomic side because I was always working on proteins majority of time. So I wanted to see how does the genomic side look like. I also wanted to share a fun fact about this character that I get like sometimes quite interesting mails. This is our lab mascot. I used it a lot for our software to promote it. And I get these interesting emails where people like write these like really formal emails asking if this here might be a cat or a monkey. They wanted to really figure out the species of this lab mascot. And I actually said it is neither of that. It's its own species. It's M.marvelous, and the name is Marv, and so it is its own thing. Just a fun fact. And before getting started, these are the amazing scientists I now have the privilege to work with. And so my lab now at the Seoul National University has this mission to accelerate biological science through fast and easy to use methods, like a really broad statement. But you will see more about what we do in the next slides. Okay, I wanted to start with metagenomics. In metagenomics, we have this ability now to study these like 99% of uncultural microbes. So what is an uncultural microbe? Let's explain first what a cultural microbe is. You can take, for example, an E. coli, you grow it in an agar plate, you can then extract the DNA from it, and then you can sequence it, and you get DNA reads out of it. This works for some of bacteria, but not for all of them. Many of them are really, really hard to grow in labs. And in order to really do research on them, you just take a sample from the soil or from a human stool, you extract the DNA from it, you have then a mix of things, and you sequence it, and then you do the analysis. So now the analysis becomes tricky because you're not working with one organism at a time, but you work with a bag of organisms at a time. So a type of pipeline here would be, you start off with samples, you have these like reads, maybe a terabyte of reads, you assemble them into contigs, like you make them longer, like you make contiguous pieces, you have 100 gigabytes or so, and then once you have them, you can start asking questions. So you can take these contigs, you can search it against databases, for example, reference genome databases, and then you can answer the question, who is in your community? For example, if something aligns very well to an E. coli, then it's most likely an E. coli. And so you can like give species label by using reference genomes. You can also search against protein databases. For example, take these contigs, extract the open reading frames, and then search them against protein databases. You then can figure out what do they do, like what kind of functions can they do. And if you really go really fancy, you can try to reconstruct metabolic pathways. So where you figure out how do these microbes communicate with each other. And you do also that by searching. You're searching these proteins against these CAC databases or UniProt databases, and you construct metabolic pathways. So the big problem is that for some of these metagenomes, especially like soil metagenomes, like environmental metagenomes, many of these open reading frames cannot be mapped back to any database. So you cannot even do anything. So you cannot even make statements. You often throw about 90% of the data away because you don't know what these proteins are doing. So how do we annotate ORs? Who's familiar with alignments? Please raise your hand if you have seen an alignment before. Bioinformatics, great. Nothing to do here. So here's a protein alignment. So I guess my next question is, do you think this is a homologous pair of proteins? Raise your hand if you think it's homologous. Yeah, a few. Who thinks it's not homologous? Yes. And then there's a lot of maybes. Okay, maybe it's always right. But so it is, when you look structurally, fairly similar. The amino acid, on the amino acid level, it's a bit hard to see, but our methods can detect it, right? Like these methods like BLAST and so on can actually align them, and you can actually find the similarity between them. But why does it work? Like, because amino acids are somehow conserved, and therefore structure stays also somehow conserved. And because of that, we can find like relationships that are really far apart using amino acid information. And that brings me to the first tool that I have done during my PhD, which was, I guess, very critical for me. It was kind of like building up the backbone for my later sciences. It's MM62. It's an ultra-fast and sensitive sequence comparison method. So let's say it's easy, similar to BLAST, but faster. And the idea of this was that you somehow needed to be fast. By how are you fast? You need some kind of pre-filtering. So if you take a protein sequence and you search it against a database, you want to find all of the possible hits, but you don't want to do a lot of like useless alignments. You only want to align the things that actually have a chance to be aligned. And so your pre-filter should reduce your big database of like hundreds of millions of sequences down to maybe 10 to the power of 4 sequences do you actually want to do an alignment with. So how do you detect an homologous sequence? So here you have two pairs of sets of sequences. Here sequence 1, sequence 2, and sequence 1 and sequence 2, one of a homolog, the other a homologous protein pair, the other one of an unrelated protein pair. And these blue dots are k-mers, short words that are shared between these two sequences. So what you can see in the homologous pair is that you can find some kind of like a trace that is in between these two sequences. And this is your alignment at the end. This is the alignment you're reporting. And so a pre-filter has to find this signal very fast and has to detect it from that noisy example on the right, because you have these like random chance k-mers that might occur everywhere. So we designed some pre-filter that finds double k-mer hits on a diagonal, does then an ungapped alignment, and afterwards does an. And so you go from a database initially 10 to the power of 8. You maybe with your first pre-filtering step, you go down 10 to the power of 5, and then 10 to the power of 3. So you in each step have to process less data. And we all optimized it very, very nicely. So now here's the point that I want to talk about. When designing MM6, I thought a lot about modules. I wanted to modularize that because I thought like a lot of the things we are doing will be reoccurring. And if we build nice modules, then we can build all of these cool methods in the future built on these methods, on these modules. So I designed one module, which was the pre-filtering module. Really just making this stage until the ungapped alignment. And designed one module that I called alignment, which does a gapped alignment of every hit that goes through the pre-filter. Old benchmarking results by now outdated. Diamond already has new versions. But here's MM6, and you could see MM6 was benchmarked against BLAST at that time, and is like as sensitive as BLAST, but faster, but it can be also much, much faster than BLAST by being less sensitive. And one thing that MM6 can also do is these profile searches, meaning you search against the database, you find hits, you use the information that you have found to make another search, and you find more hits. So in short, MM6, yeah, once we got it out, was really like a great alignment tool for you to like align proteins fast and big sets of proteins. Okay, so that work was really my initial work, and I think a lot of the ideas here came from my first supervisor, Johannes. I mean, he kind of like lined out the initial ideas, but I had the chance to contribute my own ideas a little bit. But I didn't feel like that was really all my achievement. So it was like feeling, could I do it on my own? I was not sure at that time. You know, I just saw I could build a cool system and I can make it fast, but can I really come up with an own algorithm was not clear to me. So the next one that we developed was LinClust, and I think that idea was coming mainly from me as initial idea, and that was made me really proud of it. You know, like that was the first time where I got my own algorithm, so that was exciting. And there's a linear time clustering algorithm for big sequence sets. So why is that important? So there is this review that is from 2008, really old, where Hugenholz and Gene Tyson were saying that a big problem in metagenomics is that these bigger and bigger data sets will become more and more hard to process because these all against all comparisons become really a bottleneck of this. And we see that everywhere, like all pipelines that we have in bioinformatics, these all against all comparisons are really becoming a major point that makes things slow. Because they need n-square comparisons, n-square often too slow for millions of data points. So in clustering, what can you do with clustering? You start off with a set of sequences. You then cluster them, clustering meaning you are assigning them to centroid sequences that somehow represents these other sequences. And once you have that, you actually have built protein families. You can build consensus sequences, you can reduce redundancy and so on. So clustering is often a really first step you want to do in your metagenomic analysis because you have often like hundreds of millions of proteins and you want to reduce them to a tangible size of set you can actually deal with, a non-redundant set. So at that time, clustering worked like this. So here you have on the left side dots, which are sequences. And in order to cluster them, what you did is you have these K clusters on the right side. Initially, it's empty. So you take this blue dot, and you say you're the first cluster. Then you take the next dot, and you compare it against this cluster, and you ask, are you similar enough or not? If you're similar enough, you will be assigned to the group. If not, you will be a new group. In this case, it's a new group. Then you took the next point, you compare it against this one, not similar enough. Next one, oh, it is similar enough, so you assign it to this group. And if you do that long enough, you end up with like K groups, right? So you assign at the end all of these dots to these K groups. And this is becoming like N times K, right, in complexity. And that's kind of like quadratic because K becomes really, really big. At one point, nearly as big as N, and therefore you end up with a quadratic runtime. And we checked that. So we looked at these software that can cluster these big sets like CD-HIT or uClust, and they all in runtime here had some quadratic tendency. Here on the x-axis is count of sequences in millions, on the y-axis is runtime in minutes. And so they all had these like quadratic tendencies, didn't really scale well enough. With LinClust, we then built an algorithm that can do it in linear time. And I think that really enables a lot. You can now go from 1 million to 100 million and so on, or billions, and it's not so problematic because you just scale linear in the runtime. So how does it work? What we did is we designed an algorithm around k-mers. So we go through all sequences in the set, we extract k-mers. We extract 20 k-mers, we could also extract more, but in that point we start off with 20. 20 meaning we extract them by hashing all of these k-mers. We take the lowest 20 hashes and we take them for all of the sequences. We then sort these k-mers, and now each k-mer that is the same is one of these colorful circles here that you have. So each black dot, which is a sequence that is in a colored circle, shares the same k-mer. So that's really easy and easy to compute. And next, what we do is we select the longest one to be the center of this k-mer group, like saying you're representing all of these other sequences. And that's really a really simple base assumption, like it's an assumption that's obviously wrong. So you cannot say that the longest one is the best one. But we have just made this shortcut. This is like our trick to actually become to get into linear time. And this is actually, again, going back to modules. I have built that based on the MM6 framework here, and this is an extra module. That is the k-mer matcher. This is one module we have just built. And we have this longest sequence, and now what we do is we align all of these dots against the longest sequence. We check if it fulfills the clustering criteria. If it does, then we make an edge in between them. And here we can use the next module, which is the align module that we have built before. So I could reuse my old module here already. So I had to only build one more module to make that happen, right? So it's nice. Reusability really helps a lot here. And yeah, as I mentioned, this like selection of the longest is really critical here. If you haven't done that, you need to do an all against all comparison. That's what you normally have to do to figure out what is the right group. And that is really slow. Okay, and then you cluster. And that's another module called cluster. So we are separating all of these tasks into modules. Okay, so linked cluster is great, and it really enabled these clusterings of billions of sequences, and now it's already used by many of these organizations, PDB, UniProt, Magnify, EMBL, NCPI. They're all using this algorithm because of its like scaling capabilities, mostly because they're dealing with like hundreds of millions of sequences and have no other solution to actually use something that scales linearly. Okay, now we have built a set of these modules, and now we have this Cambrian explosion of tools that comes out of it. So one tool is the Conterminator, which is a tool to find contamination in genomic databases. What it does, it uses the k-mer matcher to do a comparison across DNA sequences. It does an alignment, and then it predicts contamination based on these alignments and can figure out, is there contamination in genomes? Another tool is Metagenomic, which uses the pre-filter and the align module, and then afterwards uses some collect optimal set, which finds like a set of exons in metagenomes to figure out if something is like a eukaryotic protein. Another tool is Spacefarer, which uses the pre-filter and the alignment, and then a combined score to figure out these like spacers, these like CRISPR-based spacers. And another one is Plus, which is an assembly algorithm, which uses the k-mer matcher and alignment, and then it assembles these results. So you can see that most of them using a subset of these modules and then just add a new module on top to kind of build a new method. I want to talk a bit about Plus. So Plus is a protein-level assembler for metagenomics. It really helps to assemble these really diverse samples, like soil samples, they have really high microdiversity, and most assembly software normally fails. So Plus could actually extract protein sequences from them. And this we have done. So we went out there and we assembled like 460 soil samples and got about 2 billion protein sequences. And we assembled 755 eukaryotic marine metatranscriptome data and get 300 million proteins. And we got some extra proteins from the JGI. This at that time was the biggest protein set available. So sometimes you can actually, as a researcher with not so much resources, if you build the algorithm, you can build the biggest sets of proteins. You can make the biggest things, right? I think that's really exciting about science. So don't hold yourself back. Just go out and try to do these like big things. And this database had a lot of diversity. So if you take a CRISPR-Cas14 protein, you search it against the UniProt and that database, then you can see in blue is everything that's UniProt, in red is everything that is in this metagenomic database. So a huge amount of protein diversity that lies there. If you do these profile searches, you can find even more, but still the metagenomic side dominates. Okay, now we are at this time in history where AlphaFold, the DeepMind was working on AlphaFold. AlphaFold 1 just came out. And then what happens is we had Plus coming out, and we had also other big protein databases coming out. I got a call from DeepMind. So they were really interested in this big protein databases. Because they wanted to build this AlphaFold 2 system. So they wanted to see how they can use this data to somehow improve folding. And so why is that? Why do they need it? So because you effectively go from a small MSA to a very big MSA. Big MSA, meaning you have a bigger trajectory of the history of that protein that you want to fold. So you don't see just one sequence or 3 sequences, you see like 100 sequences. And by doing so, you actually learn a lot. And that led then to this AlphaFold, me being part of this AlphaFold team. And I guess you're all pretty much aware of the AlphaFold results, so I don't have to make it too exciting. But there's this cusp competition that happens every 2 years where they measure the performance of the structural field. So and then in 2020, the first AlphaFold system was in 2018, which is the green line here. You have these categories, easy and difficult, easy being the ones that you can solve with templates, difficult being the ones you need to do free modeling, like structures that we don't have in our databases. And so in 2020, the academic community catch up with AlphaFold. This is this blue line here. And then the AlphaFold 2 system came, right, and really changed the game of the structure prediction at that time. And we are now able to really get structures that are predicted at close to the experimental margin of error. So having really high quality structures. So to introduce briefly the structure prediction problem is the idea you start off with a sequence. You somehow have, so when you have a protein sequence and you put it into solvent, it goes down this energy funnel, and at one point it gets to this lowest point, which is the native state, which is where you have this really well-folded structure. That being mentioned, so this is really what you want to do with the structure prediction system. You want to go from the sequence to the structure that is in the native state. Again, proteins are very dynamic systems, right? So they're depending if you have ligands around and so on, they might have different conformations. But when we talk about the structure prediction systems, we really care mostly about this native state. So how did this, why did they need these MSAs? So this is a very simplified view on the AlphaFold system. So you start off with a sequence and you do a homology search. So you search against the sequence database and you build an MSA and you give that MSA to the neural network. The neural network has an MSA module and a structure module. MSA module is kind of the task to extract information from the MSA. The structure module is kind of forming that into a structure, using the information that the MSA module has extracted and turn it into a structural hypothesis. And then you loop that. So you can loop these predictions multiple times in recycles. Okay, so why MSAs? So MSAs are kind of cheating the game. So it's not predicting it from a single sequence, but you rather use evolutionary information. So you can see that you take this MSA here on the left, and this MSA kind of points somewhere in the energy landscape. It kind of gives you a hint where you have to start off your search for finding the right structure. And then the structure module is kind of refining it. It goes down this energy funnel to make it more like the native structure. And how do you generate MSAs? You take the sequence, you search against the database, you find an MSA, and then you have certain mutation patterns. And so what you can see, you have for example, these columns that are green, purely green. These are columns that you should not change, these are residues that might be functionally really relevant to the protein. Then you have this on the right side here, there's yellow, red, green areas. These are probably flexible regions where it doesn't matter so much what amino acid you put in there. And then you have these regions where you have blue, yellow, yellow, blue, yellow, yellow, blue. And these are co-evolving pieces. So you cannot change one. If you want to change one, you have to change the other. If not, something bad happens to that protein. And they're really the thing that helps you for the folding, because once you identify them, you know that these are residues that have the chance to be actually in physical contact. And if you find enough of them, the folding problem becomes not so hard anymore, because you actually constrain the problem quite a lot. So you just need to find a few long distance hits, and then your folding space, your degrees of freedom reduces quite a lot. So DeepMind knew that the MSA is important. Therefore, they put a lot of effort in the AlphaFold 2 system to build good MSAs. So they searched against multiple databases. They used a tool called Jackhammer to search against the UniRef 90, which is from the as a clustered version of the UniProt. It uses the Magnify, which is a metagenomic database containing a lot of human gut and marine proteins. And they use this BFD database that I have built for them with HHBlitz to find more homologs that they put into these MSA system. And that is really great to build good MSAs. The problem of that was, once they rolled out or they released the source code at one point, this kind of search is really slow. Actually, this search here, building the MSA, takes longer than the structure prediction that is coming afterwards. So we needed something that is faster. And here we had luck. We had MM6 at hand, right? That was our tool that we could use to replace these other search tools, Jackhammer and HHBlitz, by just using now MM6 and building a database of this UniRef 100, which is also UniProt-based sequences, and the environmental databases, BFD and Magnify combined, and then search it with MM6 and build MSAs. So we built that because we wanted to make AlphaFold widely available to the public. So we wanted to build a system where in a website you can actually put in your protein sequence and then you can get a structure prediction back. This was not, when AlphaFold 2 came out, there was no system like that. You couldn't really just do it in the beginning. So we built a tool called ColabFold. And it's exactly what I'm mentioning here. It's like effectively getting rid of that homology search and use our MM6 search to be fast for building these multiple sequence alignments given to the system. Okay. So we combined AlphaFold 2 with the Google Collaboratory environment. Google Collaboratory is an environment where you get free GPU time from Google, which is quite nice. So you can actually execute Python code on Google GPUs. And because we didn't have GPUs at hand, we couldn't really build a service where we run everything on our GPUs because we had zero. So we needed something else. So we used the Google Collaboratory to get GPUs, and we used MM6 where we built a server that takes the protein sequence that you put into this form field. It builds an MSA, it sends it back to the Google Collaboratory environment, it uses that GPU to predict the structure, and then you can download the structure. So and that's like really a web-based system that we have built, which is kind of interesting because it's not really a classical web server. It's like a web server based on Google's resources somehow. So thank you, Google, for giving GPUs to so many people. What is interesting is that this really blew up. This became really big. So we get around 60,000 requests per day now from people around the globe. We have folded over 20 million, predicted over 20 million jobs. So that's, I always compare it to K-pop videos. It's like it's a medium successful K-pop band, I would say. And that's impressive for something that is bioinformatics. Okay. So I talked about, like, I motivated this whole talk with metagenomics and not being able to annotate ORFs, right? And I've kind of completely ignored everything of this now. Now going back to this, what if we still don't find any homologs, meaning using protein sequences we don't find any homologs? What can we do now? Now we go back to this initial figure. We could, instead of using these amino acid sequences to annotate, we could use structures, right? We have now AlphaFold, we have structures available. And a good thing is DeepMind, in collaboration with the EBI, has actually predicted over 214 million structures and has made them publicly available. So we have nearly a structure for nearly every protein in the UniProt database, aside from viruses, unfortunately, but the other ones are there. And there is the ESM Atlas, based on ESM Fold. They have released over 617 million structures from metagenomic sources. So now in total, we have nearly 1 billion structures available at our hand that we can use to do something with them. And so one problem that we wanted to tackle is the problem of like annotation and like replacing homology search in a way, like classical protein homology search, but rather use structures. And that brings me to FoldSeq. So FoldSeq is this ultra-fast searching and clustering of protein structures. It's a collaborative project with the Söding group in Göttingen. And yeah, here we really try to make sense of this massive structural sets. Okay, so another question here, an alignment, 16% similarity. Do you think this is homologous? Raise your hand. It's the same amount of, it's actually more hands than before. Okay, before the identity was higher. But yeah, so structurally, they are very similar, but 16% we wouldn't really trust so much. Like in classical protein alignments, you wouldn't really trust that. Like if somebody puts that alignment in a paper, I would highly doubt that. I would not really say, oh, yeah, sure, that's probably right. But structurally, again, even visually, we can distinguish that they might be, right? And so now let's use structures instead of amino acids for alignments. And this is a really old field. Structural alignments exist since forever. So actually, structural biology might be one of the first big fields in bioinformatics. There's a lot of tools built. It has a long, old history of it. And there's two great tools. One of them is DALI, the other is STM-Align. They produce really, really good structural alignments. But they have a problem. They don't really scale to the extent of these databases. So here we have taken a SARS-CoV-2 structure, and we have aligned it against 800,000 AlphaFold predicted structures. And it took TM-Align a few days. It took DALI a week or so. And so if you think about searching through 100 million or a billion, that might take like half a year or multiple years. So not really tangible for a PhD, right? So if you have 3-4 years for your PhD, you cannot wait 3 years for getting back your answer. So with FoldSeq, we have built a system that can do this kind of search in like 5 seconds rather than multiple days, being like 4 to 5 orders of magnitudes faster. And how did we make the method fast? The idea is instead of comparing 3-dimensional objects, which is often done in structural aligners, you can much faster compare a 1-dimensional object like a sequence. And why can we do that? Because we have built this MN6 alignment framework with pre-filter and alignment. We just built a new module, right? And then we can make a structural aligner. And that's exactly what we've done. We wanted to somehow get the structure into a sequence, because then we know we can compare that very fast. So my question to you, how would you turn a structure into a sequence? Like, what's the most naive way of doing so? Reusing the amino acid sequence. Yeah, that's right. Yeah, that's right. A bit more sophisticated. Let's do one level of sophistication higher. That's the answer I wanted. Yes, that's what majority of people have done. In like there's a field before that have turned structures into sequences, and they mostly focus around the backbone torsion angles, like really the adjacent amino acids, and kind of turn it into secondary structures, or like more sophisticated secondary structures by using more states to represent them. But from a search perspective, that isn't so great, because imagine so if you turn it into a secondary structure, it means you would take that helix and it will turn to H, like a long stretches of H's, right? You kind of lose information there because you only know there's a helix. You don't know that the helix is next to a sheet, right? But if you would knew that, then you could maybe leverage that information and make a better alignment choice. Because a helix alone occurs everywhere in the AlphaFold database. It's just you take that helix and you find it like in 100 million entries. So it's not really helping your pre-filtering step very much. So you don't gain any speed. So we wanted to build a structural alphabet that encodes the nearest neighbor information, like the information about some interactions, because that's really what structures are about, interactions with other residues. So here we're starting off with this like red residue here. That is the residue you want to encode. We find a nearest neighbor in three-dimensional space, which is this like bluish part here, and we extract the angles around this nearest neighbor and the angles around the residue you want to encode, the distances in between them, and we encode that as a feature vector, which we then discretize into 20 letters. So from a pipeline perspective, nearest neighbor in structural space, what does that mean? If you just imagine the nearest neighbor is often quite a boring one because it's like you adjacent left or adjacent right? That's not really so meaningful. So we put out these like virtual centers, which look into space like 45 degrees from the C alpha to the C beta. And we use these virtual centers to identify the nearest neighbor, which is the blue one here. Okay. We then extract these 3D descriptors, and this is a vector, and we put it into a neural network, which is an encoder network, which turns this vector into a two-dimensional vector, which is then assigned a letter to. Like you have a dictionary in the middle that you use for assigning these letters. And you train this like encoder by using a decoder. This is also called variational autoencoder, or here specifically VQVAA, a quantized version of it, where you use the input and you try to predict the output from the input. So the input and the output is the same. You just go through an information bottleneck in the middle, and that kind of like forces the network to learn a lower representation of these vectors. So it's a really great way of clustering vector space. So whenever you deal with vector space and you want to like cluster it down, think about VQVAE, so just variational autoencoders. Okay, so that's how we construct our alphabet. Our alphabet is called 3DI. So let's go back to this initial example. So here you have the amino acid similarity of 16%. And what happens now if you switch out the amino acid information with the 3DI information? If you do so, you get a sequence that is way more conserved. So you can actually now align these sequences. You have like 54% sequence identity, and you can also estimate the similarity between them more reliably. Okay, so we have benchmarked fold-seq against these other structural aligners using domains. Here's the plot for the SCOP superfamilies. SCOP superfamilies are domains that are in a relationship that's quite far apart. So most amino acid aligners cannot detect superfamily membership very simply. It's like quite difficult. And you can see that because MM6 is here. Like you can see this amino acid 1, and this is this orange curve. And the recall is not that great. So you don't really find a lot of these like really distant connections. While with structures, you can actually find a lot more. And these structural aligners all perform fairly good. So you have the TM align there, DALI, as well as fold-seq in red, and they're all in a similar range of performance. But keep in mind that fold-seq is about 4 to 5 orders of magnitudes faster. And so I wanted to, I mentioned that about metagenomics, like how can we use structures for metagenomics somehow? And there's a group in Canada, Greg Glor's group, and what they did is they do research on wastewater phages. So they did long-read sequencing of wastewater phages, and then they extracted these ORFs, open reading frame from these wastewater phages, and they tried to annotate them. And they're really recapitulating my initial case. When using classical pipelines like Bacta, which uses HMM-based comparisons, they could map back about 10% of these ORFs to reference databases. So really, again, this is the case where we would throw away 90% of the data. But with collab-fold, like first predicting the structure of these, and then using fold-seq to map against the structural databases, they could map up to 60%. So really increasing the amount of information that you can get about these really divergent phages. Another cool example is from the Benfield group. They looked at these giant bacterial machineries. So they are these machineries that have over 80,000 amino acids. Like it's a single ORF with 80,000 amino acids in bacteria. This is bigger than Titan. So it's bigger than every biggest protein we have in human. And they wanted to dissect and understand what are these big ORFs are doing. And they use classical methods like HMM-based. So they cut these pieces, these big ORFs into pieces. And they use classical methods like HMM-based methods, mapping back against databases, and could find quite a lot. But for every position where you can find one of these like pink donuts, that's regions where they use collab-fold in conjunction with fold-seq to map them back because these HMM methods could not resolve anything. So I think it's really great to see that it has impact on metagenomics already. And you can use it for annotating really divergent things. And you can easily use it by search.foldseq.com, which is our web server. You can put in a PDB sequence, structure, sorry. You can select all of these databases. You can search through them. Hopefully it doesn't take too long. Normally it should be in the ranges of seconds. If it's too long, let us know. And you can click through the databases. This, for example, the CAS database. You can see the 3-dimensional alignment here on the right side and the amino acid alignment on the left side. Okay. But to be like truly on the level of metagenomics, we need to be faster. ColabFold needs about a few minutes for, in combination with AlphaFold, needs a few minutes for a structure prediction. In metagenomics, we deal with millions or even hundreds of millions of proteins. So predicting these take really, really long. So here's really where Michael Heinzinger's work come in. So what he has done, he has made a translation system from amino acid to 3DI. You can imagine that going from English to German or any kind of like bilingual system. So he trained a transformer model that takes in these amino acids and gives you out these 3DI strings. And it can do this kind of predictions 4,000 times faster than if you would do a structure prediction first and then encode into a 3DI string. So this really like shortcuts you, right? So you don't need the structure prediction at this point. And you can use it out of the box. We have built it in FoldSeq. So with these commands, you can just give it and then a FASTA file, and it will do the searches for you using FASTA file inputs, but internally using structures by using this PROST-T5 model. And performance-wise, this PROST-T5 model in our benchmark is comparable with real structures. So it predicts these 3DI strings as good as you would get that actually from the ColabFold-based prediction or from experimental structures. You can also try it out. If you want to try it out and see if you don't trust it, you can go to the web server. You can put in an amino acid sequence. You push this predict button here, and then you can get a 3DI prediction, and you can search against these databases without using any structures. And you can find then like these different databases again, and you can then see the alignment, but you don't see the blue structure anymore. The reason for that is we don't have the three-dimensional coordinates. We only have the 3DI string. We don't have like a full structure. We just use the 3DI string for searching. But it's much, much faster. So maybe a good way for annotating things where you mainly care about the annotation. You don't care so much about the structure. Okay. So in summary, like FoldSeq discretizes structures, these tertiary traits into like an alphabet of 20. It's fast. It's about 4 to 5 orders of magnitude faster than these structural aligners. It has accurate e-values. So meaning if you are familiar with BLAST e-values, you can transition to FoldSeq fairly simply by just trusting e-values in a similar manner. They are well calibrated. It produces local alignments. So it means you can find domain rearrangements where you have like AB and like BA. So you can find these two different domains. It's open source, and it was published in 2023, which is, I wanted to talk a bit about it. Publishing takes really, really long. This one took insanely long. So the first version of FoldSeq was out in 2021. We put the first web server out and the source code. We then submitted a manuscript, I think, in early 2022, and it took like 1.5 years to be accepted. And actually, in 2024, it went into print. So it took about three to 4 years to get that really out. So my suggestion for you guys is put out your code and let people use your software as early as possible. And don't hold back because a journal needs really, really long for reviewing. So put out the source code, make a preprint available, like talk about it on social media and so on, and make it clear that people know what you have done. And so that's really good, because in this case, it took really long. And without that, people couldn't have used it until 1 year ago, which I think would be a big waste of opportunity. Okay, the last thing I want to talk about is clustering the AlphaFold database. So what we wanted to do is, we wanted to go from 240 million to amount of clusters, and clusters being here groups of machineries, like structural machineries, where more or less the domain structure is preserved across the whole chain, and we wanted to put them into groups. Normally here I make a comparison between clustering runtime, but you learned about clustering. Clustering takes n squared time, right? So 214 million times 214 million would be a really big number and would be even too slow for FoldSeq to do. But the good thing is we have lint-clust, right? So we learned how to reduce that to a linear comparison time. And for lint-clust, we used sequences, and we have sequences. We can use the 3DI sequences now, which represent structures. So by combining these both ideas, we could just reuse the old modules and build something new, right? A structural alignment, a structural clustering pipeline. And that's what we have done. We took these 214 million proteins, We clustered them with MM-seq down, and then we used full-seq clusters, and we clustered that into like about 18 million clusters. So going from 214 to 18 million, but many of them are singletons. We rejected singleton clusters. We ended with like 2 to 3 million clusters that are non-singleton clusters. This is a near factor of 100 in reduction in size, right? So, meaning that we can already make a statement. There are about 2 to 3 million machineries in these clusters, like where at least the domain composition is the same, and structurally they're somehow similar to each other. And we then looked at these like 2 to 3 million clusters, and this we have done together with Pietro Bellatro's group at the ETH. And that was published also last year. And we looked at these clusters from multiple perspectives. One is like these darkness, like we wanted to figure out how many of these clusters have no annotation at all, and we don't know really what these things are. We wanted to understand how do these clusters distribute in evolutionary space, like what kind of machineries are universal to all life, what are like eukaryotic machineries, and so on, what are species-specific machineries. And we wanted to look at domains to see if we can use domains to annotate other domains. So the first thing is this darkness analysis. We were actually really, really stringent. So what you can see here on the left side is the AFDB with 214 million entries, and actually very little of them are not annotated, meaning most of them have an annotation. And the reason for that is we have what we have used as an annotation criteria is if you take the structure and you can align it back to PDB, it's annotated, which is obviously not a great annotation, but it's at least some kind of annotation. And if there's any kind of P-farm annotation available, it's annotated. If there's a tiger farm, it's annotated. So in the AlphaFold-DB itself seems very little annotated, non-annotated under our criteria. But if you look at these clusters, these 2 to 3 million clusters, 35% of them have no annotation at all. And what that means is also no member in the cluster have any annotation. So we really checked that neither of them has one. So one trend is known proteins tend to cluster together. And that somehow makes sense, right? We have these like big families and they somehow collapse into these groups. And so these big groups, we know what they are. The small groups, we do not know what they are. And you can also see the same trend if you look at the amount of members. If it has 2 to 4 members, it's about 40% dark. And the more members it has, the less dark it gets. But it's a bit of a deceiving plot. There are some groups there that have thousands of members that are universal to all life, and we have no clue what these things are. So I think that's a great starting point to investigate experimentally what do these things do, right? These dark machineries. We looked at these clusters in taxonomical space using lowest common ancestor analysis. Who's familiar with LCA? Please raise your hand if you have heard about LCA. Not so many hands. Okay, then let me explain LCA. So what we did is we took a group, and let's say in one of our clusters, there's a member from a eukaryote and a member from a bacteria. We now trace back the tree to where do these two things meet. Eukaryotes, bacteria, cellular organism. Eukaryote will be eukaryote, right? And more bacteria. And like only E. coli. So it means like the more universal the group is, the more it is on the left side, the more specific it is, the more it is to the right. So you have about 530,000 that are universal to all life. Those are machineries that are at least in 2 super kingdoms. You have about 371,000 that are bacterial specific, meaning they are important to bacteria so that they end up at the bacterial node. And interestingly, 311,000 that are eukaryotic. So bacteria to eukaryotes have a very similar ratio in machineries that are universal to them. I would have expected to see way more bacteria because bacteria seem to have shorter generation times. And actually we have way more bacteria in our databases. But that's what came out of this analysis. And then if you go more to the right, you actually have some species-specific structural clusters, meaning these are clusters that have a structure in it that is only from the same species, but has at least 2 members. And I think they are a mixture of reasons why you have them. Some of them are really like technical problems you have in your analysis, where you recall the same wrong ORF twice from an E. coli genome, and they turn into the same wrong structure, and you put them into the same cluster. But there can be also some interesting cases in them. So we haven't looked really, really deeply in them, but I think there are cases where you can probably find new emerging genes potentially. We looked at interesting links, right? People always want to find something interesting. So what is interesting? It's always like probably people will say, can you look at the data and find something interesting, right? So what we wanted to do is like we wanted to find evolutionary links that we wouldn't expect so often. So going from human, going to bacteria, using machineries that we wouldn't expect to see in bacteria, for example. So starting off with histones. So we have histones in humans, and we looked at the cluster of histones, and we couldn't find histones in bacteria. And there's actually a field of research now where people have figured it out before. There are histones in bacteria. People try to figure out what is the function of them. It's not clear. We have this innate immune system in humans, and it's obviously interesting to see if that exists also in bacteria. Here is a structure that detects bacteria, and it then causes cell death. And we can find it in human. It's known in human to do this, but it's also in bacteria. It's also actually previously detected, so it seems to be also working in bacteria in a similar manner. But something that we find novel is this AIM2 gene, which does something similar than the middle one, but it detects viruses. And you can structurally also find it in bacteria, and we can find also auxiliary machinery needed for that function. So I think it's something like a great case to investigate if it also has the same AIM2 function in bacteria. Okay, there's much more to explore. You can go to cluster.foldseq.com. You can put in any kind of UniProt identifier, and you can explore the clusters. They are also now in the AlphaFold database. You can also find these cluster members in the AlphaFold database. You can filter these members by taxonomy. You can click on any of these members. You can see how they compare to the representative of that cluster. And you can find similar clusters at the bottom. They might have different domains attached to them. For example, here you can see that the yellow part is an extra domain that is only the blue one was shared, but the yellow one is an extra piece of domain. So in conclusion, I'm coming from this world where we use amino acid information to make all of these kind of predictions that really drives bioinformatics, protein interactions, functional residues, and so on. I think we can really supercharge this analysis using structures. So we have really these big resources at hand, and we have now a lot of opportunities here. And going back to this like modules idea, I think it's really important for you to start building your trusted code base. You have now in your PhD the time to build cool things, and you can build them in modules, and you can reuse them over time. And the more of you cool small things you build, the faster you will be able to build bigger new things on top of that. So make building small modules, make that your backbone. That's a trusted code you can reuse. And maintain it. Make sure it functions. Reuse it in many projects as possible. The more you reuse it, the more you will find bugs that you can fix. Set up continuous integration. So that's why I was very excited about the unit tests. Use tests. Test your software. Make sure that it actually functions. If you figure out something, try to make it better and better over time. And package your software well. Make it sure that people can install it easily. That's really a major hurdle for most bioinformatics tools to be used. So make it that it's as easy as possible for people to use. And then more people will actually use it. And with this, I want to thank everybody involved in the projects, and I want to thank you for listening.
Thank you, Martin, for that amazing talk. And as a structural biologist, thank you for FoldSeq. I also think we have some time for questions. So if anyone wants, I can.
Yes, thank you. So, of course, big takeaway from the talk is make modules that you can reuse. And so I wanted to ask the question in the development of each of these modules, did the development of new modules, was it a process of saying, oh, we have these new tools, we have MMSeq2, the next logical step is to make FoldSeq with this? Or does it start in the other direction? Does it say, hey, we have this need for something like FoldSeq. Oh, I actually already have all these tools here.
Yeah, it's the second version of it. So we have built MMSeqs in the initial idea already around the idea of building modules. So these modules were like the pre-filter and alignment, maybe some result filter, maybe some like create DB module. And so similar to like these GitHub, GitHub add, GitHub commit, GitHub. So you build these, and like once you have them, you see more opportunities how to use them like broadly. And then if we wanted to build FoldSeq, which I think about, we have all of these modules. How can we like orchestrate them to make this tool happen, and how much new things do we actually have to develop, how much old things can we already reuse how they are. And so I think that's the way how we think about this. Yeah. Thank you.
Thanks. Do we have order?
Okay, so I have a conceptual question and a technical question. So for the technical question, so when I try to reproduce the FOSIC paper result, I find that the DALI result is very different, probably because I use DALI Lite and you use vanilla DALI. So where do you get that copy?
The copy from DALI, you mean?
Yes, because I can't find anywhere.
The version that we have used, we have written the version string in the paper, but I can help you if you need to. We can also figure that out. But there, we always write the versions that we have used in it. But yeah, if you have a problem, I'm happy to help.
Okay.
Yeah.
Okay. So for the conceptual question, when you build BFD, you always remove the singleton sequence in the metagenomic sequence, even though they may still contribute to a better MSA for structural prediction purpose. So what is the rationale of excluding singleton sequence?
So our rationale is that gene calling is not perfect. Gene calling has a high sensitivity, but a position around 90 or so. Like you have an expected 10% of wrong ORFs. So that means they accumulate. So if you get databases with the database have like now 30 billion, or like even 40 billion proteins. If you keep all singletons in, your database become insanely big, and it became with a lot of noise, right? The singletons were really noisy at the end. So we rejected them because practically it makes our searches really slow. It needs way more resources if people want to download it and so on, and providing it to the world. And I agree that there are probably singletons in them that are interesting and good for the MSAs, but like it's like a cost-benefit trade-off that we have done. Yeah.
Okay, thank you.
I think we have time for one last question.
I'm the present, you are the past.
Gonzalo, I will answer you later.
So mine's somewhat technical, and maybe you can answer for the room. So I know it's been superseded by Prost T5, but in the original CAE you had for developing the 3DI thing, in your CAE you went down to 2 dimensions and then expanded out to, you had a dictionary. Mm-hmm. Was there a reason technically? I know most of the people in the room at some point are probably going to try and write some sort of model to do something similar with a CAE down to a low dimensional representation. Was there some reason you went down to 2 and then expanded to 20 instead of having the CAE, the encoder, actually go to 20 dimensions and then go from there? Or like, so is there some insight there that is missing maybe?
Yeah, it's a good question. We ended up by adapting a VQ VAE, and we have optimized it to work in the best way. We have optimized hyperparameters on our training set. I assume one of them might be this, but it's a good question. I think Michael has done most of the optimization of the VQ VAE, so probably it's better to ask him. But I think we started off with like a very default way how to use it, and it was like you have to go to a latent space that is lower dimensional, right. And T2 seems to be good, and then you cluster them and you make k-means on top of that. If you want to just go to 20 directly, yeah, I'm not sure if it would even converge to nice discrete states, or you would just get more blurry over time. So I think that might be another problem that might occur there. Yeah.
Cool. Thanks.
Thank you. I think that was all the time for questions. Or if it's a quick question, Gonzalo, you might ask.
But
no, it's fine.
Only a great question.
No. First of all, I want to say that we might be in the presence of the biggest star of our generation. I want to say that. I witnessed all these transitions because we shared the retreats. You were never in Göttingen, but we shared the retreats. Now, my question is, how do you envision now that the structure, the sequence structure gap has been closed, and you have all these good methods? What's the next step? And I guess in terms of functional notation, and I think you are also already working a little bit in quaternary structure. What are the next challenges, especially because here there's a lot of students that are starting their careers. So if you could be now starting your PhD, where would you invest your time?
Oh, good question. So I think my career advice is stay away from hype. Don't jump into hype because it's a meat grinder, and I think that's a bit can be a bit unhealthy. And you know, this is mostly only like a few winners there, and really hard. I would always think about where data is going. So so when building like MMCX2 and Lindlust, we saw like there's more and more proteins, and you saw on the horizon like there will be billions. At that time they were like millions, right? So you saw that you needed something for these like billions. And I think the same thing I'm thinking now, for example, conformations for protein structures, right? Like, would it be cool to build a tool that makes sense of conformational space? Because that will be probably something we might be able to predict at a certain level in the near future, and then we will have like millions of them. So always think about what is like the next thing rather than the current, right? Like where does data go, and then think about what you can build that makes that analyzes this data. Then I think that's how what I would do it when I do a PhD again. Yeah.